So I'm John Lambert. I've joined ImmunoGen as the second scientist on a project that was funded by venture capitalists who formed the company ImmunoGen, but started it at the Dana-Farber Cancer Institute as an incubator space. And from 1981 until 1987, that's where the ImmunoGen research started. And I joined in 1982, in fact. Then in 1987, we became a real biotech, and I've been at the company for now nearly 34 years. I think after the approval of Kadcyla and Adcedris, which suggests that, in fact, ADCs can work. I mean, this was the culmination of nearly 3 decades of effort. It's clear that actually now targeting chemotherapy to cancer cells can be done by antibodies to yield active agents that have good side effect profiles, very benign side effects. So that was, of course, the object of all of our research, was to have more effective drugs with less cytotoxic side effects of classic chemotherapy. And so now that's been achieved, I think that it's unleashed a whole new wave of research to identify the right targets for this technology, and even the new technologies to approach different targets. So I think the world of ADCs, the future of ADCs in cancer treatment is going to be quite a broad one. The most interesting work, I think there are 2 areas. One is now, I think, really understanding actually what targets are the right ones, and also how to measure target expression, because this is targeted delivery of chemotherapy, and I think understanding how to find the patients that express the target that will respond is one key area. So I think that's one area. The other is these potent tubulin agents, maytansinoids and the auristatins, are clearly going to be a valuable class. There'll be more than 2 agents with these payloads approved. But actually there are many cancers that don't respond very well to tubulin agents. For example, colon cancer, notoriously, the tubulin agents were never active. And just because you're adding the payload to an antibody to deliver it, it doesn't change the underlying sensitivity of the disease to the mechanism of action. So I think the new developments with DNA alkylating payloads, other types of payloads that interact with topoisomerase, for example, I think now people can see it can work. I think a lot of groups are digging in to now expand the types of mechanism of action in the payload class, so that's exciting. I think many cancers that are underserved, but I think the future growth, it will be in colon cancer, lung cancer, these major cancers. It's to identify the right targets for have active ADCs against solid tumours. So I think there's a lot of room for growth, actually. There's a lot of unmet need. The challenge is, in the end, it has proven easy to make something that will kill cancer cells upon antibody binding. I think the challenges are to get the therapeutic index right. And so, following the premise that at best, a few % of your antibody will ever bind to a tumour cell. So one always has to remember that greater than 90% of the ADC that you administer systemically will always be metabolized by normal tissue, so you hope that the rate of which that metabolism is slow, because antibodies have a long half-life, and the metabolites formed can be dealt with and eliminated from the body safely at a dose that allows efficacy as well. So it's to actually get the toxicity right. And the field has learned that the animal models for toxicity, be it mice, rats, even non-human primates, have not actually been particularly good at assessing what the dose-limiting toxicities will be in humans, and even what the doses that you can achieve will be. And so while it's exciting to be developing new payload classes, until one has some human clinical data on the tox profile of these new classes, it's still a little bit of an open question whether there will be a therapeutic index. It's easy to do that in mice. It turns out to be not so easy in humans. So I think there are two exciting things I can think of. One will be for some solid tumours. I think there'll be some ADCs that will move into clinical trials that could prove to be pivotal, on which approval might be expected if the clinical data hold up. So that'll be exciting to actually have some more approved products in solid tumours. And I think from more on the clinical research side, I think the emerging ideas of combining ADCs with the new exciting immunotherapies, especially immune checkpoint inhibitors, that ADCs may be ideal agents to combine with these immune checkpoint inhibitors, especially since the ADCs spare the immune system from the penalties of chemotherapy, which knocks down dividing immune cells, the very cells that immune checkpoint inhibitors want to actually stimulate. So I think ADCs and immune checkpoint inhibitors could be very, very interesting combinations, and I know many people have talked about this at meetings. I think in the next year these ideas will start to emerge in the clinic.